Bismillahirrahmanirrahim. My dear second student, we are now starting with the histology of urinary system. As you know, that the urinary system comprised of kidney, ureter, urinary bladder, and the urethra. Kidney, ureter, urinary bladder, and urethra. The main function is production of urine and excretion of waste, and also act as a endocrine gland, excretion the very important renal enzyme in the blood circulation. And the components of the urinary system, the mammalian urinary system, comprised of kidney, remove urea and excess water from the blood. The ureter transports urine from kidney to the urinary bladder, and the bladder, which is the storehouse of the urine, and the urethra, which transports urethra from bladder to the exterior. Kidney, ureter, urinary bladder, and urethra. So this is the broad functional aspects of the components of the mammalian urinary system: kidney, ureter, and urinary bladder, and urethra. Now, if we will first discuss the kidneys are two bean-shaped organs. Mean, that is the oval in shape, located in the left and right side of the abdominal cavity in the retroperitoneal space in the paracolic gutter. In adult, they are about 11 centimeter, 4.3 inches in length, as this diagram will depict, because the 11 centimeter, then the width is about 5 to 6 centimeter, and breadth is about 2.53 centimeter. The kidney receives the blood from the paired renal artery, which are the branches of the abdominal aorta. Blood exits into the paired renal veins. The blood from the kidneys drain into the vena cava by the renal veins. Each kidney is attached to the ureter, a tube that carries excreted urine to the urinary bladder. Now, the internal gross features of the kidney. Now, this is the diagrammatic presentation of the cut section of a kidney. Look at the visible feature, which you can see by the naked eye. This is the hypothetical diagram, and this is the actual cut section. Compare it. This is almost similar in appearance. The outermost is the cortex. Outermost is the cortex, then is the medullary region, which is innermost. The cortex invades in the form of a renal column. In between the medullary renal pyramid, that move upward in the form of a triangular shaped conical, having apex. The apex open into the minor calyx. The minor calyx leads into the major calyx into the pelvis. Now, this is the outermost portion, is the cortex. Then is the renal pyramid, which is triangular. The base is toward the cortex, whereas the conical apex is toward the minor calyx in the medulla. The minor calyces, 1 or 2 or 3, they will drain into the major calyx and then into the renal pelvis. Now, these renal pelvis lead into the ureter. Now, look at this diagram. The outermost is the rim of the cortex, and then is the triangular shape is the renal pyramid. The renal pyramid open into the minor calyx, then major calyx, then the renal pelvis. This is the ureter. In between the renal pyramid, you see this is the renal column. These are the blood vessels of the kidney, the renal artery, and renal vein. Now, this is the cut section feature you can see by your naked eyes. Now, what is the histological functional unit of a kidney? Is the nephron. It is the structural unit. Look at the picture. 1 to 1.5 million in adult kidney. In an adult kidney, it is about roughly 1.13 million in an adult kidney. They consist of nephron. It consists of the Bowman capsule, which containing the duct of capillaries from afferent and lead efferent arteriole. The Bowman capsule is a cup shape which encompass or encircle the capillary plexus in the glomerulus. This is the glomerulus. This is the Bowman capsule. The Bowman capsule leads into the tubular system, proximal convoluted tubule, then the thick loop of Henle, thin loop of Henle, again ascending thick loop, and then is the distal convoluted tubule leads into the collecting tubule. These collecting tubule ultimately drain into the ducts of Balini, and then in the apex of the renal pyramid, from where into the minor calyx. Now, this is the structural unit. It's perform all the functional aspects of the kidney. You will have to memorize their components. Now, what is the cell histology of the kidney? Where these cells are located, and we will have to see all these in coming discussion. Renal histology studies the macroscopic structure of the kidney. Distinct cell types in the histology of kidney includes the kidney glomerulus, parietal layer, then is the kidney glomerulus podocyte, basal layer. So glomerulus, the Bowman capsule consists of two layers, the parietal layer which line the Bowman capsule inside, whereas the basal layer which approximate with the glomerulus duct of capillaries is called the podocyte, which is specialized cell. Then the kidney proximal flush border cell. These are the cells which line the proximal convoluted tubule, loop of Henle, thin segment cell, which is famous type. Then thick ascending limb cell, kidney distal tubule cell, collecting duct principal cell. Collecting duct is the intercalated cells, and interstitial cell, kidney cell, which are the mesangial cell, and other cells are also located within the kidney parenchyma. You will have to look at the various types of cellular lineage present within the parts of the nephron. Now, this is the histological picture broadly. A Chinese stain you see in a light macroscopic picture. Now, this one is the outermost is the cortex, consisting of the Bowman's capsule, proximal and distal convoluted tubule, multiple Bowman's capsule with duct of capillaries over here you see, and multiple proximal and distal convoluted tubule, renal corpuscle. Then is the medullary. These are the medullary rays. You look at the picture. So these are consisting of the loop of Henle and collecting ducts visible in the medulla. Medulla is the longitudinally running tubule, loop of Henle and collecting tubules. Whereas in the cortex you see invariably convoluted tubule, proximal and distal, and Bowman capsule and duct of capillary, that is the glomerulus. So these vertically running tubular system is called as medullary rays. Now we'll discuss with the first is the glomerulus. What is that? So glomerulus is a tuft of capillary, that is the branching of the afferent arteriole, which is capped by the Bowman capsule. Look at the picture. So this hole is the Bowman capsule. Now this is the afferent arteriole carrying the blood toward the Bowman capsule. So it will divide multiple capillaries, and these capillaries they fill the space within the Bowman capsule, and it is lined by the itself endothelial, but outermost is the podocyte, which is the visceral layer of the Bowman capsule. Now from this, after purification or given the various solutes and absorbing some solvents or many other micronutrients, it's again the blood capillaries unite to form a afferent arteriole. So this is the efferent arteriole, that is the exit arteriole. This is the afferent, that incoming arteriole carrying the blood from the renal artery, and this move toward the efferent arteriole from it after purification within the renal glomerulus. So all in between these capillaries and podocyte, you see multiple cells of the, which are called as mesangial cell. So what is the overview sheet diagram? The juxtaglomerular apparatus. No, this is the location of a juxtaglomerular apparatus. Look at the picture. Afferent arteriole, JG cell is the modification of tunica media cell, which become neurosecretory. It is the endocrine part of the kidney, from where there is a modification of tunica media of the afferent arteriole. Look at the picture. These are the GJ cell, become the secretory cell, which create the renal. Now this is the modification Macula Densa cell in the distal convoluted tubule. Distal convoluted tubule, the cells, they become modified and become sensitive to the various changes, the solute and solvent. So they will give the sensory input via the adrenergic fiber and give appropriate response to secrete the renin. So this whole system, along with the mesangial cell, afferent arteriole, JG cell, and the macula densa and mesangial cell, these all constitute the JG apparatus. This is very important apparatus, which is concerned for the controlling of the body fluid and also controlling the blood pressure of the human being. So look at the picture. The lining is the simple squamous, which is the parietal layer, and the podocyte cell, which are the visceral layer of the Bowman capsule, which directly imposed on the capillary plexus, so it formed the filtration barrier, which we discuss in coming slides. Now you will have to memorize over here the JG cell, JG apparatus, afferent arteriole with the JG cell along with the macula densa cell with mesangial cell in the vicinity. Now renal corpuscle and the filtration membrane. What is the mechanism, what is the anatomy of the filtration process, and what are the structure which form the filter between the blood and the urine. Now look at this picture over here. This is the afferent arteriole, which carries the blood toward the Bowman's capsule. So over here it forms a multiple closed system of tubular network, which is called the capillary plexus. These capillary plexus having continuous basement membrane, but its endothelial is fenestrated. Endothelium is fenestration, but it has a continuous basement membrane. Now this is the cavity in the Bowman capsule. It's lined by the simple squamous epithelium. This simple squamous epithelium is the endothelial lining. It is also called as the parietal layer of the Bowman capsule. This one you see multiple branches, cytoplasmic pseudopodia of a podocyte. This is the podocyte nucleus, the cytoplasm. These are tentacle or multiple pseudopodia which approximate with the capillary lining, forming the wedge-shaped structure and can pass the whole capillary lumen. Concentrate on this. This is a podocyte nucleus. Look at the picture, and this podocyte nucleus send the primary secondary processes, that is the pseudopodia, which encircle this lumen of the wall of the capillary. So there will be interval between the tertiary pedicle of the podocyte. These intervals are actually the membrane interval from which the filtration will take place. So this interval you will have to memorize. It is between the foot processes or pedicles, tertiary pedicles of the podocyte and the basal membrane of the capillary, which give these filtration state. Again, this diagram you see This is the nucleus of a podocyte cell body. This is the primary pedicle, secondary pedicle, and tertiary pedicle, and a proximate endothelial lining of the capillary, and only the interval remain between the capillaries and the podocyte. These intervals are the filtration slits. These are filtration gaps and through which the urine is produced by the ultrafiltrate. Now these are fenestration mean pores between the endothelial lining. Basal membrane is continuous. Capillary, glomerulocapillary endothelium, podocyte covering, and basal membrane removed over here. Now to look into the pore complex in the fenestry is between the endothelial lining, although the basal membrane is continuous. Now these are the four processes of podocyte: primary, secondary, and tertiary. Now these tertiary interdigitate with each other of the podocyte processes when they interdigitate, but there is a relatively gap, and this gap is called as filtration slits. Now these are multiple small gaps. These gaps are the filtration slit, which play a key role in the formation of urine by ultrafiltrate. Now the Bowman capsule is a cup-shaped sac at the beginning of the tubular component of a nephron. The glomerulus capillary loop is from the afferent arteriole and collected within the Bowman capsule. The vascular pole is the side with the afferent arteriole enter and the efferent arteriole exit. So always remember, you have seen the diagram, that one pole is afferent and efferent arteriole, another pole is proximal convoluted tubule. The proximal convoluted tubule is the urinary pole, whereas the afferent and efferent arteriole is the vascular pole of a Bowman capsule. The afferent arteriole carry blood from the renal artery toward the capsule, where efferent leave the capsule. The urinary pole is the side where the proximal convoluted tube starts. The layer of Bowman capsule are as follows: a parietal layer, a single layer of simple squamous epithelium that line the capsule. It does not function in filtration. ये मैंने पहले आपको बता दिया कि ये वो लेयर है जो कि इनसाइड के सिंपल स्क्वेमस लाइनिंग है, पैराइटिस बनाती हैं। बॉमन कैप्सूल का कोई रोल नहीं है इसका इन द फिल्ट्रेशन ऑफ द ब्लड to form the urine. The visceral layer lies just above the thickened glomerular basal membrane and made up of podocyte. Visceral layer is a specialized podocyte cell. Beneath the visceral layer lie the glomerular capillaries, just approximated with the glomerular capillaries. These specialized cells having a cell body, primary, secondary, and tertiary pedicle or pseudopodia. They will encircle the capillary plexus in the basal membrane. Rather, they join or share the basal membrane and they encircle the capillary plexus. They are actually the very important podocyte. They are concerned for the filtration of the blood to form the urine. Now look at the picture. It's a hypothetical diagram. This is look at the envelopment of the podocyte. Structure of the podocyte, star shape, having multiple pedicle or pseudopodia encircling the blood capillaries, forming a filtration slit. This is the cytoplasmic along with nucleus, primary pedicle, secondary pedicle, tertiary pedicle, and multiple small branches arise and invaginate or approximate with the capillary basal membrane, only give a small slit-like intervals. These are small slit-like interval in between the processes. So these processes are the filtration sites. ये वो जगह हैं जहाँ पर filtration होती है, because all the rest is the approximated. Only this part is the area where the urine formation will take place by ultrafiltrate of the blood. Structure is the cytoplasm with nucleus, primary, secondary, tertiary, and multiple branches of the pedicle that will encircle the blood vessel to form a filtration slits. What is the filtration barrier? between the visceral and parietal layer into which the filtrates enter after passing through the filtration slit. What is the filtration barrier? The fenestrated endothelium of the glomerular capillaries, the fused basal lamina of the endothelial cell, and the podocyte, the filtration slits of the podocyte. Now these are which is covered by a thin, which is covered by this filtration slit of the podocyte is covered by a very thin diaphragm. Now always remember the three or four structures which are involved in the filtration barrier: fenestrated endothelium of the glomerular capillaries, fused basal lamina, endothelial cell podocyte, and the filtration slit, the gap, which is covered by a thin diaphragm. So these are the known as the filtration barrier. Now what is the basal lamina? How it is formed? Now the basal lamina consists of three parts. Lamina rara external. It consists of adjacent to the podocyte process. Now this is actually the basal membrane of the podocyte itself. What is that composition? It is formed by the heparan sulfate, and it blocks by charge of the various types of charged particle. उसको block कर सकते हैं. Lamina densa is the dorsal central zone. So this is the central zone. What? That is the fused part of the basal lamina of this podocyte along with the endothelial lining of the capillary plexus. It is made up of type 4 collagen and laminin. Block the size of the molecular weight and up to 70 kilodalton. Blocks the size below this, can pass through it. ब्लॉक्स द चार्ज पार्टिकल. कोई भी चार्ज पार्टिकल लैमिना रहा उसको रोकेगा, लेकिन जो दरमियान वाली जोन एंड थिक जोन वो 7 किलो डाटा तक वो रोकेगा, उससे बाकी जो है बिलो वो पास कर जाएंगे. लैमिना रहा इंटरना एडजेसेंट टू द एंडोथेलियल सेल. दिस इज द basal membrane, which is the endothelial itself membrane. It is made up of heparan sulfate. Block the charge particle. चार्ज पार्टिकल जो पास थ्रू नहीं करेंगे, तो दरमियान वाली जो है डार्क सेंट्रल जोन द लैमिना डेंसा, इट इज एक्चुअली द ब्लॉकिंग साइट फ्रॉम वेयर नो वन कैन पास above the 7 kilodalton. Now this is the diagrammatic electron microscopic picture along with the diagrammatic presentation of a slit membrane. Now look at the picture. Now this is the lumen of the capillary. This is the endothelial lining. So this is the endothelial lining. Look at this is the fenestrations of the endothelial lining. Over here the endothelial lining is absent, but the fused membrane is barrier. Now this is the fused basal membrane, the basal membrane of the endothelium, the basal membrane of the podocyte. This is lamina densa, lamina rara internum, lamina rara externum. These are the multiple tertiary podocyte pedicles. Now this is the gap gone to multiple gaps. These gaps are called slit membrane. So this slit filtration slit is covered by a very thin diaphragm. So podocyte cell body, the tertiary processes which approximated with the basal membrane of the endothelium, giving a very small opening pore. This is called a slit process or slit membrane. This gives way for the formation of urine. It is covered by a diaphragm. Now this is the diagrammatic presentation of a filtration barrier. This is the endothelial lining. This is nucleus of endothelium. This is the fenestry of the endothelium. So this is the fused basal membrane of the endothelium toward the capillary endothelium of the podocyte. So this is the thick basal membrane, lamina rara externum, lamina rara internum, and lamina densa. Now these are the put processes of the podocyte approximated to the capillary basal membrane. So this is this filtration slit. This is the picture of a filtration slit. Over here you see thin diaphragm, slit diaphragm. So slit diaphragm. So what are the structure which present between the blood and urine from podocyte toward the anterior? Podocyte put process, slit membrane, diaphragm, fused basal membrane, endothelial lining with fenestrae, and then the lumen of the capillary plexus. These are the structures which form a barrier between the blood and the urine. So you will have to remember all these structure. ये आप लोगों ने मुकम्मल याद रखने हैं कौन कौन स्ट्रक्चर जो हैं दरमियान में आते हैं ब्लड और यूरिन की फॉर्मेशन में, और कैसे स्लिट मेम्ब्रेन या स्लिट डायफ्राम बनता है उसको ध्यान में रखना है. वेरी इंपॉर्टेंट फॉर क्लिनिकल एस्पेक्ट्स, इसको आपने फौरन ध्यान में रखना है ये बातें. Now what is the proximal convoluted tubule? Now next move is the portion of a duct system of a nephron which leads from the Bowman capsule to the loop of Henle. Lining क्या है? It is the cuboidal epithelium with brush border. The microvilli greatly increase the lumen surface area of the cell. The cytoplasm cell is densely packed with mitochondria which are largely found in the basal region within the infolding of basal plasma membrane. Extensive lateral interdigitation between the neighboring cell. क्या स्ट्रक्चर इसमें होंगे? cuboidal epithelium लाइन करेगी, apical surface microvilli, basal infolding and lateral interdigitation. All these are the cells which are concerned for ion transportation. Very, very rich in the mitochondria for energy provision of the cell. Hence they are staining with the eosinophilic dyes. Cuboidal epithelium lumen is having the brush border, more microvilli because of the increased surface area. Lateral interdigitation, again the surface area increase, interdigitation between the neighboring cell. Mitochondria are forming. Now this is the proximal convoluted tubule, Henle stain. Look at this is the basal membrane, this is the one cell, this is the nucleus, rounded nucleus. The lumen is the brush border. Numerous microvilli giving the surface area to be increased. Cytoplasm is eosinophilic, granular in appearance because of the mitochondria and plasma enfolding. Longer hence are more numerous in the cortex. ये लंबी भी हैं और cortex में ज़्यादा हैं. Contain a single layer of cuboidal cell with eosinophilic granular cytoplasm. Cell boundaries are not distinct because one cell is not distinct because of the lateral interdigitation. आपस में जुड़े हुए हैं इसलिए उनकी बाउंड्री जो है वो इतनी विजिबल नहीं है, but basal membrane is present. Cell having the macula on the luminal surface to typical brush border. Luminal side पे जो है brush border appear होगा. This is the ultrastructure of a proximal convoluted tubule, that is EM structure. Look at the picture. This is the plasma enfolding, numerous mitochondria toward the basal side. Apical is the microvilli, full of microvilli. This is the lumen, the nucleus, nucleoli, mitochondria, plasma enfolding, lateral interdigitation. So this is the typical picture of a ion transporting cell in the proximal convoluted tubule. Distal convoluted tubule is the portion of kidney nephron between the loop of Henle and the collecting ducts. The DCT is lined with the simple cuboidal cells that are shorter than those of the proximal convoluted tubule. Distal convoluted tubule is lined by a simple columnar same cell, but this is the shorter in length. Those of proximal convoluted, the lumen appear larger in DCT than the proximal convoluted tubule. पहला फीचर जो डिफरेंशिएट करेगा लार्जर ल्यूमन है लेकिन इसकी लेंथ कम है. ब्रश बॉर्डर इज रिलेटिवली लेस एज कम्पेयर टू प्रॉक्सिमल कंडक्ट, ऑल्दो इट इज प्रेजेंट बट इट इज रिलेटिवली लेस, कम होगा एज कम्पेयर टू दी प्रॉक्सिमल कंडक्ट ट्यूब्यूल. DCT can be recognized by its numerous mitochondria, basal unfolding and lateral membrane interdigitation with neighboring cell. Same picture. Distal convoluted tubule also a ion transporting cell, so it has the same picture as that of the proximal convoluted tubule, except lumen is larger, less in microvilli, presentation brush border, and length is relatively small as compared to the proximal convoluted tubule. What are the difference? This chart will tell you the difference between the proximal convoluted tubule and distal convoluted tubule. Apical brush border not present usually in the distal convoluted, although present relatively less if present, but brush border apical is usually present in the proximal convoluted. Eosinophilicity more less. cytoplasm more less. Readily discernible nuclei less likely, more likely. Nuclei are more prominent as case is to the distal convoluted tubule. Now this is the H&E stain of a high powered microscope to differentiate the proximal and distal convoluted tubule. Look at the proximal tubule. The nuclei are not so much prominent, but the lumen is having full of microvilli, but the distal convoluted relatively less. The nuclei are more prominent. So this is the picture diagram of a proximal and distal convoluted tubule. What is the loop of Henle? Is the portion of a nephron that leads from the proximal convoluted tubule to the distal convoluted tubule. It meant for the concentration gradients of the various solute and solvent. It is present in the middle of the kidney. The tissue type of loop of Henle is simple squamous epithelium. Loop of Henle can be divided into three parts: thick descending limb of loop of Henle, thin loop, and thick ascending. You all know that the three parts: thick descending, thin loop, thick ascending. Lining is the simple squamous epithelium, concerned for the concentration gradients according to the solute and solvent, and it is present in the medullary rays of the middle of the cut section of the kidney. The collecting duct system of the kidney consists of a series of tubule ducts that physically connect the nephron to the minor calyx. So always remember that is the nephron जो उसका distal convoluted tubule है उससे connect करेगा with the minor calyx. Each component of the collecting duct system contains two types of cells: intercalated cells and a segment-specific principal cell. So two types of cells are नजर आएंगे collecting duct system: principal cell and intercalated cells. The principal cell mediates the collecting duct's influence on sodium and potassium balance via sodium channel and potassium channel, located on the cell's apical membrane under aldosterone effect. So the principal cells जो हैं collecting duct के उनको हम कहेंगे meant for the sodium and potassium pump, because they will maintain the equilibrium between sodium and potassium via sodium and potassium channel pump. It is under the influence of aldosterone hormone. Integrated cells, these are the cells comes from the alpha and beta varieties and participate in the acid-base homeostasis. So it is concerned for the acid-base mechanism, controlling mechanism, homeostasis, and these are the cell of alpha and beta variety and participate in the acid-base homeostasis. Now these are the collecting ducts, and look at the picture. So collecting ducts are concerned for the equilibrium of this duct system, the equilibrium between water and solute and solvent, and also concerned for the under the influence of aldosterone maintain the sodium and potassium level, and they are also concerned for the acid-base balance. And this is the brief summary of the histology of a nephron loop. Look at the picture. So this is the Bowman capsule, parietal layer, simple squamous, visceral layer, that is the specialized podocyte, the slit membrane, fenestrated and basal lamina. Then is the proximal convoluted tubule. So this is the proximal convoluted tubule histology, basal enfolding, apical macrovilli, nucleus is pushed toward the surface. Then is the distal convoluted tubule. This is again present, plasma enfolding, mitochondria prominent, few macrovilli. Then is the loop of Henle, simple squamous epithelium. Collecting duct is the tall cuboidal cell with the nucleus. These are also concerned for the various solutes and solvent transportation, containing much more mitochondria as well. So this is the summary of the histological lining of the cell types: the Bowman capsule, proximal and distal convoluted tubule, the blood vessel pattern. Now this is the collecting ducts you see in this diagram. Now this is the DG apparatus. Again I will summarize. Now look at the picture. So this is the Distal convoluted tubule, lining is cuboidal epithelium. So this is the modification of this lining epithelium become the Macula Densa cell, which become tall cuboidal. So they are the neurosensitive cell. They will sensitize the sodium potassium ions and give information of the concentration values as well as the volume of the blood to the higher center. Now this is the JG cell. These are the cells which are the changes in the tunica media. Tunica media consists of smooth muscle, you all know, in the arteriole, but this modified smooth muscle become JG cell, which are secretory cell, and they are concerned for the excretion of the renin enzyme, and these are the extra glomerulus mesangial cell, and these are capillary plexus, and this is the parietal layer, and this is the visceral layer lining the capillary endothelium. So concentration is on Macula Densa, JG cell, and interstitial mesangial cell. Component क्या है? Macula Densa cell specialized lining the distal convoluted tubule at the afferent arteriole, from where this will sense the sodium chloride concentration within the blood. JG cell, they are specialized smooth muscle mainly in the wall of the afferent arteriole, some in the afferent arteriole, and the cells that can they synthesize, store, and secrete the enzyme renin. This glomerulus cell harbor B1 adenine receptor. When stimulated epinephrine or norepinephrine, the receptor induces the secretion of renin. So JG apparatus is very digitally innervated by the beta-1 adenine receptor. Release of epinephrine and norepinephrine, these receptors induce the secretion of renin, which is when released within the blood, it will invariably controlling the volume as well as the blood pressure of the body. So this is the histological picture of a JG cell. Now look at the picture. So it is the afferent arteriole, tunica media having the modified JG cell in the picture you see. This is the distal convoluted tubule. This is the Macula Densa cell. The lining of the cuboidal epithelium is changed into the various types of specialized cell, which is known as Macula Densa cell. Now these are the JG cell and macula. This is the picture of a typical picture of a glomerular apparatus. This is the afferent arteriole, so this is the distal convoluted tubule. The rest is the Bowman Mesangial cells also visible. This is the electron microscopic picture of a JG apparatus. Now look at this is the renin secreting granules present within the cytoplasm of the this afferent arteriole tunica media. This is a typical picture. These are the secretory cells present within the tunica media of the afferent arteriole. So this is the electron microscopic picture of a JG apparatus. Now look at the Macula Densa. These are the modified epithelial cell. Look at this is a tall cuboidal epithelium. It is more granular in shape. It is neurosensory cell. Changes cells in the distal convoluted tubule. The lumen of distal convoluted tubule, rest in the cuboidal cells are present, but these are the modified Macula Densa cell. Along with the previous slide, you see it is the renin secreting cell in the afferent arteriole and the tunica media. Now, this is a mechanism you will very much will be taught in the physiology as well as biochemistry. So what is the role of renin? So in this diagram you see the renin is released on stimulation by the adrenergic receptor. The renin is, when present in the blood, it will activate, stimulate the liver angiotensinogen, convert into the angiotensin 1. The angiotensin 1 when circulate in the lungs and the endothelial lining, where the angiotensin converting enzyme is present, it will activate into the active substance angiotensin 2. So angiotensin 2 will activate the sympathetic activity, also stimulate the renal gland, produce the vasoconstriction and release of ADS secretion. So it will all mechanisms which control the increase in the volume as well as the controlling the blood pressure. So angiotensin 1 and angiotensin 2 converting enzymes are present within the lining of the lung. That is the type 2 alveolar cells. So all you know, this is the coronavirus is also having a target on this AC converting enzyme. alveolar type 2 cell. So over here the angiotensin 1 is converted by AC receptor and release the enzyme, and then the angiotensin convert which is active hormone. It will influence the sympathetic activity, solute particles, adrenal gland, aldosterone secretion, then vasoconstriction and adhesion from pituitary, ultimately increase the volume and this conserve the sodium ion to increase the blood pressure to face the fight and flight of the body. Now, what are the mesangial cells? Are specialized cells in the kidney. They make up the mesangium of the glomerulus. They are present in the vascular pole of the renal corpuscle. 30 to 40% total cells in the glomerulus. So it is present, majority of the cell present within the glomerulus, the mesangial cell. The primary function of mesangial cell is to remove the trapped residues and aggregated protein from the basal membrane, thus keeping the filter free of debris. Very important, a very crucial role. That is the wear and tear mechanical effects produced within the filtration process. The residues and various types of unwanted protein when get collected within the filtration slit, it will be absorbed and engulfed by mesangial cell. Mesangial cell have irregular shape and flattened cylinder-like cell bodies. Having actin, myosin, actin and filaments give mesangial cell contractile properties, because these cells because actin and myosin, actin, they have the contractile property. So they are concerned for the scavenger cell. They remove the debris within the filtration areas. Now this is the methodology, role of the mesangial cell within the glomerulus. This is the mesangial cell present. So it is the not specifically concerned because it is more physiological as well as biochemical. So you will have to memorize only that is it has a relation with the capillary as well as the glomerulus as well as the role in the filtration mechanism of the kidney. So only you will have to memorize its role, that is the phagocytic activity within the glomerulus. This is the clinical aspects. So these are the glomerulonephritis is the very known picture you see in the clinical aspects. Nephrotic syndrome, nephritic syndrome, glomerulonephritis, all these are due to the diseases present within the glomerulus, in the filtration slit, in the filtration membrane, basal membrane, and the mesangial cell activity. So this is the clinical aspects you will have to study in the various books. Hematuria, proteinuria, and RBC are the clinical picture of the glomerulonephritis. Usually it affects the glomerular area in the basal membrane, filtration slit, the podocytes, and the mesangial cell. Now this is the pictorial presentation. This is normal. Look at the abnormality. These foot presses become fused. The basal membrane itself becomes fused with the various inflammatory cells. So there is the blocked or choked filtration slit leads into all the solute become back within the blood, leading to hyperuria and urea syndrome and chronic and acute renal failure will take place by these action of these various types of the various types of the inflammatory cells. Thank you very much. Next we will move on to the ureter in the next lecture.